This lecture is about the cardiac system, part 2, in which we're going to get into some physiology. So first, to really understand the function of a cardiac muscle cell, we have to remember its anatomy. Here you'll see a slide of a cardiac muscle cell, and if I said to you, what are some characteristics of a cardiac muscle cell, you might say, well, probably the clearest indication are these little dark lines, which are called intercalated discs. So only cardiac muscle cells have intercalated discs. The other thing you'll notice is that a cardiac muscle cell is defined as the distance between one intercalated disc to the next. So this is one cardiac muscle cell. Now remember, for skeletal muscle cells, they would stretch the entire length of this slide. So they would go from one end to the other and keep going. They're long and thin. But cardiac muscle cells are actually short and wide. The other thing you'll notice is that besides the dark vertical line, there are other lighter vertical lines here. Those are called striations. So cardiac muscle cells have striations. They also have one or two nuclei. Now the nuclei are a little hard to see. They look kind of like they're pushed to the outside of the cell, which they are. So we would say they are uni or binucleate. Another characteristic is that the cells sort of branch. Now remember, skeletal muscle cells were side to side, they're parallel. But you can see right here, if we look, it looks kind of like a Y here, the cells are actually branching. And so that's a difference between cardiac muscle cells and skeletal muscle cells. The cells branch. Lastly, this isn't something you can see under the microscope, but cardiac muscle cells are under your involuntary control. So you cannot consciously control the contraction of cardiac muscle cells. Let's take a closer look at these intercalated discs to see what's going on there. And if we do, we would see a couple of things. One, you would see connective tissue in there, which is called the fibrous skeleton of the heart. We talked about that in cardiac anatomy lecture number 1. But the other two things you'll see are these kinds of junctions. The first is called a gap junction, and we talked about this in A&P 1. But in this slide, this is the plasma membrane of one cell. So that cell, this is the cell here, and then here's the plasma membrane of another cell, and you'll notice the intercellular space here. But you'll also notice there are these structures connecting the cells. They're kind of like tubes, and these are called connexons. So these connexons, what they do is they allow ions to rapidly move from one cell to the next. Ions. Now remember that ions have a hard time getting through the plasma membrane because of that lipid bilayer, phospholipid bilayer. So this allows ions to pass quickly between cells. Now, why would they want to do that? Well, one of the major ways that cells communicate to each other is by passing chemicals back and forth. And so these connexons are actually involved in cell-to-cell communication. And this occurs between cells, these gap junctions. You see them when cells have to communicate very quickly. And in fact, cardiac muscle cells have to communicate very quickly. Think about the atria. Both atria have to contract at about the same time in order to pump the blood to the ventricles at the same time. That means that every single cardiac muscle cell has to contract pretty much at the same time in both atrias. And they do that by getting the message through the gap junctions. Okay, so quick cell communication. Another thing you'll see between cardiac muscle cells and in this intercalated disc area are these desmosomes. Now desmosomes are also between plasma membranes, so Here, this gray line here is the plasma membrane of one cell, okay? And that cell is over here, if you can imagine that. And here's the plasma membrane of another cell, which is over here. And you'll notice again there's a space between the two plasma membranes, the intercellular space. So this structure is the desmosome, and there's a couple of parts to it. One is this purple kind of button-like thing. This is called a plaque. And think of it as a block of cement. Now coming out of this plaque are these things called intermediate filaments. And you probably talked about these in biology. Now the intermediate filaments, what they do is on the other side of the cell is another plaque. And there are other intermediate filaments that are connecting kind of like this, okay? So, you know, if I said to you, and it's on this cell too, what does it look like its function is? You might say, well, think of these intermediate filaments as steel rods. So you've got a hunk of concrete here, another hunk here, and these steel rods between them. They're actually working to reinforce the cell. So what this is doing is it reinforces the cell itself. And that's because if the cardiac muscle cell is going to be pulled on either end, because when cardiac muscle cells contract, they pull inward. And remember, it's attached to another cardiac muscle cell here and on the other side. And so this one's pulling inward, this one's pulling inward, and it's being pulled on the other side like this. So again, the cells could be torn apart if they weren't reinforced by these intermediate filaments attached to the plaque. Now the other thing you'll notice is between the two cells are these structures called linker proteins. And linker proteins do what they kind of sound like. They're linking the two cells together, they're holding them together. Because not only are they being pulled, each cell is being pulled on either side, but the cells are pulling away, trying to pull away from each other as they contract. So the linker proteins are actually going to hold adjacent cells together. And that's pretty important because when cardiac muscle cells contract, they do so with incredible force. So if you did not have the desmosome reinforcing each cell and holding adjacent cells together, the tissue would pull apart. So, cardiac muscle cells can produce a tremendous amount of force, much, much more force than skeletal muscle cells. And in order to understand that, we need to look at the structure of each cell, because remember, structure determines function. So, the first thing we're going to look at is what initiates contraction. What is the first step in contraction of a skeletal muscle cell? Well, it's a nerve impulse, or better known as an action potential. And remember, this action potential comes from the nervous system. Comes from the nervous system. In a cardiac muscle cell, the first step in contraction is also an action potential. But this action potential does not need the nervous system. It comes from cells within the heart called autorhythmic cells. Okay, so these are the heart's own action potential generators. So there's a difference. Another difference has to do with the amount of ATP stored in the cell. In a skeletal muscle cell, there is about 2% ATP by volume, 2%. In a cardiac muscle cell, there is about 25% ATP by volume. So right off the bat, you can see, oh, cardiac muscle cells produce more force because they have more stored ATP in the beginning of the contraction. They can contract longer. And so they produce much, much more force. So if we were going to give a 1-up arrow to the skeletal muscle in terms of force, would give 2-up arrows to the cardiac muscle. Another important thing is how they generate ATP. So for skeletal muscle, of course, the best way to generate ATP is aerobic respiration. But they can also revert to anaerobic respiration if necessary. Now remember, aerobic respiration, you get about 36 ATP per glucose molecule. Anaerobic is not as efficient. You only get 2 ATP per glucose molecule. So not as efficient. Cardiac muscle cells only use aerobic respiration. Only use aerobic respiration. So they're only using the most efficient method. Okay, so again, why do cardiac muscle cells produce so much force, so much more force than skeletal? Right here you can see, first of all, they store a lot more ATP because they're using a much more efficient process to make it. But there's another reason which is equally important, and in order to talk about that, we have to look at the action potential. So in AMP1, we went over action potentials when we talked about the muscular system. And this is just a quick review. So if you do find this confusing, I recommend you go back to your textbook and go over the generation of action potentials in the nervous system and also in the muscular system. But this is just a quick review to try to remind you. So this is supposed to be a cardiac muscle cell, this round structure here. And you can see that this is the plasma membrane, the dark black line. But you can see we've got negative charges on the inside of the plasma membrane and positive charges on the outside. So all cells at rest, not just cardiac muscle cells, even skeletal muscle cells and nervous neurons, they all have an overall negative charge on the inside of the plasma membrane and an overall positive charge on the outside of the plasma membrane when they are at rest. The membrane is said to be polarized. Polar means opposites, right? So negative and positive are opposites. Now these negative and positive charges are actually generated by ions. And remember, an ion is a charged particle. So I've drawn some ions in here. We've got potassium, sodium, which has a positive charge, chlorine, negative charge, and the big A stands for negative ions, and negative ions are called anions. So it's just a general term for all those other anions in there. But you'll see you've got the same ions outside the cell as you have inside. So think of it this way, if you add all the ions up on the inside, you end up with an overall negative charge. If you add the ions up close to the plasma membrane on the outside, you end up with an overall positive charge. And this is what generates these differences, which makes the cell polar. There are different concentrations of the ions inside and outside. Now, of course, the positive ion wants to go where the negative is, and the negative wants to go where the positive. The other thing is that ions, like other chemicals, always go down their concentration gradient. They want to go from where they're in a high concentration to where they're in a low concentration. Potassium is higher inside than outside, so it wants to move down its concentration gradient to the outside. Sodium is higher on the outside than the inside. So these ions want to move, but they're kept apart by the plasma membrane. So the separation of charges across the plasma membrane results in potential energy. Remember, potential energy is stored energy. And if you put a voltmeter with a little probe in, you can actually measure this potential energy, which is measured in volts. So when the cell is at rest, this energy across the membrane is called the resting membrane potential. Now remember that ions can only mainly move across the membrane through channels. And you can see that we've got the plasma membrane here, the phospholipid bilayer, and these things wedged in the middle, these are just proteins. But the proteins are acting as channels, cell transport processes. There are different types of ion channels wedged in plasma membranes. One type is called a chemically or ligand-gated channel. And you can see here, these only open up when a chemical binds to it. And we talked about this in AMP1 with the neurotransmitter acetylcholine and how it binds to a chemically gated channel and lets sodium in and potassium out at the same time. which causes an end plate potential. Other types of channels will open and close when the voltage changes. So when the voltage, let's say the charge across the membrane is minus 90, maybe when it gets to minus 85, this channel opens and lets sodium in or out. Other channels are always open. They call these leaky or leak channels, just a little, and they allow ions to dribble in and dribble out, just like if you had a boat with a hole in the bottom and water was dribbling in. And there's another type of channel called mechanically gated channels. These open and close in response to pressure, but we're not going to talk about these here. Okay, so when it comes to an action potential in a cardiac muscle cell, they are mainly controlled or caused by voltage gated channels opening and closing at specific voltages. There are different types of voltage gated channels based on what ions they let in and out. So there's sodium voltage gated channels, there's potassium, calcium, chlorine. The action potential, what happens is when the voltage gated channels open and they start to let in a positive charge, it's going to change that negative on the inside to become less negative until eventually the inside of the cell becomes positive. And so an action potential is a large depolarization that results in a reversal of charge inside and outside the cell. It is self-propagating because if we look at this voltage gated channel, this is letting, let's say that this is the inside of the cell here. Okay, so as it lets, if it opens up and lets sodium in, it makes the rest of the membrane a little bit more positive. So if here it was minus 90, as you let in these sodium ions and they start to move along the membrane, now all of a sudden we're up to minus 85. That opens the next voltage gated channel and that lets sodium in. And that, the more sodium that comes in, the inside of the membrane becomes minus 85. That opens the next voltage gated channel. So eventually what happens is it's self-propagating. When one opens, the next and the next and the next, and you get what's called a nerve impulse or an action potential. So again, this is a quick review. If this doesn't make sense, I would go back to the muscle chapter and the nervous chapter in A&P 1 to review. But let's look at that action potential. So this is an action potential, a graph of one, of a cardiac muscle cell. And the x-axis here is time in milliseconds, and the y-axis is voltage in millivolts, so it's the charge across the membrane. Now when the cardiac muscle cell is at rest, the resting membrane potential, the charge across the membrane, is minus 90 millivolts. That's what it is. So the cardiac muscle cell just kind of hangs out until something happens, some stimulus happens. Now the stimulus is actually going to be the transport of ions from another type of cell, which is next to the cardiac muscle cell. And I'm going to tell you right now that's called the autorhythmic cell. Now we'll talk about this later. But the stimulus is ions get passed. These are positive ions that get passed into the cardiac muscle cell. So if it's at minus 90 and you start getting positive ions, it's going to go to minus 89, minus 88, minus 87, and at some point it opens a voltage gated channel. Now the type of channel it opens is called a fast sodium channel, and that's because when these channels open, they trigger the opening of many others very quickly, many other sodium channels. So you end up with a rapid depolarization. Now depolarized means it is no longer polarized. So instead of being minus 90, it goes to minus 89, 88, 87, 86, all the way up to plus 30, super fast. Okay, this is kind of important. Now when you get up to plus 30, perhaps the sodium channels close because they open and close at certain voltages. So the fast sodium opened, I don't know, somewhere around minus 87 millivolts. It closes at plus 30. Another channel opens up at plus 30, and that's going to be potassium. Now when the potassium channel opens up, it lets potassium out. Okay, so when this channel opens up, it lets potassium out of the cell. So now it's losing positive charge, and you can see where it's going from plus 30 2 plus 29, 28, 27, that triggers the opening of slow calcium channels. Now when these channels open, they let calcium into the cell, okay? So now you're letting positive charge out, potassium, but positive charge in, calcium, at the same time. And so you get a plateau because it's a net zero change, right? You're letting out positive charge, but you're letting it in too. So nothing changes for a while. Slow calcium channels close slowly. So it's like closing a door slowly. So eventually they close. And when they do, no more calcium gets in. But the potassium channels are still open, so you lose positive charge. And you eventually go all the way back down to where you started, at minus 90. This is an action potential, okay? And once this happens, you'll see the muscle contract, the muscle cell contract after that. Now, why is this important? Well, let's look at an action potential for skeletal muscle cell. And here it is. We've got the same time scale and voltage. Now you'll notice that a skeletal muscle cell is at rest at minus 85. So that's the millivolts. So it's a little different, but pretty similar. So when you look at these two, what's the first thing that you notice? Probably the first thing you notice is this one's a lot longer. It's a lot bigger. Basically, it takes a lot longer, okay? This one has a pretty rapid depolarization, as does this. So we could say they're both pretty rapid, although believe it or not, cardiac muscle is very rapid, okay? Repolarization is when you go back down to negative, where the cell is polarized. So you can see that repolarization in a cardiac muscle cell, a skeletal muscle cell, is fairly rapid. So I'm going to say rapid. Okay, it goes down pretty quickly. But repolarization in a cardiac muscle cell is a lot longer. It's got a slower decline. And that's because of this thing called a plateau. So this is the plateau. Plateau is like, a flat top of a rock or a mountain. And the reason it's slower is because of the slow calcium channels. Now, skeletal muscle cells do not have slow calcium channels. They have calcium channels, but they're not slow. So they don't get this plateau. Okay, so now here's an important difference between skeletal muscle and cardiac muscle. The cardiac muscle has a longer action potential. Okay, so we're going to say it is longer and skeletal muscle is shorter. And actually we know the numbers. So a skeletal muscle cell's action potential is like 1 to 2 milliseconds, and in a cardiac muscle cell it's somewhere between 200 and 300 milliseconds. So it's 100 times longer. Why does that matter? The longer the action potential, the longer the contraction. So the cardiac muscle cell has a longer contraction, much longer, than a skeletal muscle cell, shorter. And the longer the contraction, the more force it generates. And so that's why we would say a cardiac muscle cell generates at least twice as much force as a skeletal muscle cell, okay? So the bottom line here, again, is we're trying to figure out why cardiac muscle cells produce so much more force. And one reason we said earlier on is because it stores a lot more ATP. So it can contract longer, producing more force. But the other reason is it has a much longer action potential because of these slow calcium channels. And as a result, it contracts longer. The longer it contracts, the more force it generates. And so we can summarize that right here. Again, the longer the action potential, this is force down here, the more force it generates. And so this is an important point because early on in the study of the heart, people were having heart attacks. Scientists thought, well, maybe we can take a little patch of skeletal muscle cells from somebody's gastrocnemius, which remember is the muscle on the posterior lower leg, and just patch it into the dead part of the heart, so you can think about the heart doesn't really look like this, but let's say these are you had a heart attack and these cells, this little patch here was destroyed. So some people thought, well, we'll just take the skeletal muscle cells and put them in that patch. But they found out that the skeletal muscle cells could not keep up. They couldn't generate enough force, and it just didn't work. And now we know why that is. Okay, so again, generation of this action potential, what happens next? Well, the same thing that happens in a skeletal muscle cell. So this is just from the skeletal muscle chapter. This is a neuron. Of course, you wouldn't have this in a cardiac muscle cell, but once you get an action potential, the same thing that happens in a skeletal muscle cell happens in a cardiac muscle cell. The action potential zips down the T-tubules. It triggers the terminal cisternae to release calcium. Calcium binds to troponin, which pulls tropomyosin off the binding sites, and the thick filament can bind and do its power stroke. And then the sarcomere shortens, and eventually the whole muscle cell shortens. So again, I just put this here as just a quick reminder, but if this doesn't make sense, you should go back and look at that muscle chapter. Now, I just want to mention there is a cardiac worksheet that I put up on Canvas, and I really recommend you go through and see if you can fill it out. And one of the questions is, explain to me 2 reasons why a cardiac muscle cell can produce as much force as a skeletal muscle cell. And if you'd like to fill that out and send it to me, I will look over your answers, because these are the kinds of questions you could have on an exam, and it's good to be able to explain it. Okay, so let's take a step back now. We're going to look at the heart again, and there are 2 types of cells in the heart. The first type is cardiac muscle cells, and 99% of the heart is made up of these cells. Some people also call them contractile cells, so you might see this in the text. Don't be confused. Their role is basically to pump the blood from the heart to all the tissues of the body. Okay, that's pretty much the role of that. 1% of the cells in the body are called autorhythmic cells. 1%. And some people call these pacemaker cells. Okay, these are not muscle cells. They're similar, but they cannot contract. Okay, they're modified muscle cells. And their role is to produce and initiate an action potential, independent of the nervous system. So these are responsible for making sure the heart contracts in an orderly manner. Atria first, then ventricles. So again, these autorhythmic cells are called pacemaker cells. They are connected to cardiac muscle cells by those gap junctions, and they initiate an action potential. Some people call these action potentials pacemaker potentials, just to differentiate them from the ones made by the nervous system, but they are just action potentials. So again, they ensure that depolarization and contraction occurs in an orderly manner. So the question is, how do these action potentials work? And you can see I've written down here, we now know, but let me show you in this graph. So this is the graph of the action potential of a pacemaker cell. And you can see the x-axis is time in milliseconds. The y-axis is membrane potential, charge across the plasma membrane. And here, these autorhythmic cells, when they are at rest, the membrane potential is minus 60. Okay, just a little different than cardiac muscle cells. Remember, they were minus 90. So what happens in these autorhythmic cells is they have these leaky channels. Okay, so they have leaky channels. And these leaky channels let in sodium. So again, think of a boat with a tiny hole in the bottom. Water is always bubbling in, little at a time. So these channels are letting in sodium a little at a time. Okay, these aren't fast sodium channels, they're slow. So sodium comes in, which is a positive charge, and you start to drift from minus 60 to minus 59 to minus 58. And when you get up to minus 40, the slow sodium channels close. Remember, it's a voltage gated channel, opens and closes at different voltages, but another voltage gated channel opens. It's called calcium. Now this is not a this is not a slow calcium channel like you find in cardiac muscle cells. It's just a regular old calcium channel, and this lets calcium in. And so now you get a lot of positive charge, and you get up to about +2, +3. That closes the calcium channel, but at that voltage another one opens, potassium. Potassium leaves the cell, positive charge leaves the cell, down, 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 all the way back to where you started, -60. Now, when this happens, this action potential, positive ions are passed on to the neighboring cardiac muscle cells, and that opens the fast sodium channel, and that starts off their action potential. These autorhythmic cells keep depolarizing over and over and over again, forever and ever, until you die. So this is how these autorhythmic cells work. These are the ones that generate action potentials in the heart. You don't need the nervous system. So if you, there's a little video, a couple of slides, the next slide actually, which I'm going to recommend you look over, and it's going to go through the cardiac action potential and the autorhythmic one. But how does the action potential pass? So here is an autorhythmic cell. So it's and then these are also supposed to be cardiac muscle cells. Okay, so you'll notice in the cardiac muscle cells here, you can see the thick and the thin filaments. That's what these are supposed to be. There's a nuclei. Okay, so these are all cardiac muscle cells. This part here that you see between the cells is supposed to be the intercalated disc. Okay, now remember, in this disc you've got connexons, desmosomes, the fibrous skeleton of the heart, but here they're trying to focus you on these tubes, which are the gap junctions containing the connexons. So autorhythmic cells are connected to cardiac muscle cells by gap junctions, and cardiac muscle cells are attached to the next cardiac muscle cells by gap junctions. And so when we look at this whole process, the autorhythmic cell starts first with the leaky channels, generating action potential, positive ions then, like sodium, potassium, calcium, they pass through the gap junctions to the cardiac muscle cell. And what that does is triggers the fast sodium channels, right, which then close and the potassium channels open. Then the calcium channel opens, you get the plateau. Calcium closes, you're back down. So again, the action potential that comes from the autorhythmic cell is triggering the action potential in the cardiac muscle cell, and that's kind of how it works. So this is kind of tricky, this part, and what I would recommend you do is watch this video. This is from Mastering A&P. It puts it all together. This is another video from another company that you can look at, but I hope this will help.